In this presentation, I'll talk about a survey paper on LLM-assisted clinical trial recruitment. LLMs have made impressive breakthroughs in applying medical knowledge. This graph shows the performance of models on MedQA benchmark, where recent models achieve 96% accuracy. LLMs are also popular in clinical decision-making tasks, including clinical trial matching. Clinical trials progress in multiple stages in increasing pools of human populations. They form an essential part of evaluating drug efficacy. Yet, they are time and resource intensive. Each clinical trial can take several years before reaching completion. Given the constrained budget, selecting the required patient size and population is crucial for a trial completion. This forms one of the major bottlenecks, with various estimates saying that 80% of the trials fail to recruit on time, and around 50% of the failed trials terminate because they fail to recruit enough participants. In this survey, we aim to answer 3 questions. How are LLMs used in trial recruitment? How are they evaluated? And what are the critical challenges in LLM adoption? We conducted our survey between 2019 and May 2025, and we opted for Google Scholar as a search tool for broad interdisciplinary coverage. We used a combination of keywords to search for terms related to both LLMs and trial recruitment. We then filtered out candidates that were topically unrelated, duplicates, did not have full text available, and were not related to the recruitment pipeline components. We ended up with 27 papers. We additionally included papers via citation networks of 2 well-known trial and patient matching methods, Jin et al. 2024 and Warnow et al. We ended up with 52 papers in total and covered a diverse set of publication venues across medicine, computer science, and interdisciplinary publications. Our survey is segmented into the components in the trial recruitment pipeline, beginning with data sourcing, followed by information extraction, matching patients and trials, and finally, evaluation. Prior work surveying this topic is limited in its scope and look at a handful of studies. We start with data sourcing. It comprises clinical trials. These are extensively documented via trial plan, their location, the investigators, and more importantly, for the task of matching, participation criteria, a set of inclusion and exclusion criteria that must be satisfied to qualify for participation in the trial. Trial descriptions for publicly funded trials are available in online registries. Clinicaltrials.gov is one of the largest public repository of trials from all over the world. Data sourcing also includes patient records, and these come from heterogeneous data sources, including structured tables of medications, billing data, demographics, rich unstructured text from clinical notes and discharge summaries, and image reports such as those obtained from radiology. Since patient data contains sensitive information, there are strict policies on sharing and using patient data. Mimic is one of the largest publicly available EHR data from emergency patient admissions across 8 years. Synthetic EHR data is often created for research and public sharing. Usually, pharma companies and trial centers work on private EHR data. We will look into 2 public benchmarks used by majority of the papers we surveyed, the 2018 N2C2 cohort selection task and the recurring TREC clinical trial tasks from 2021 to 2023. The N2C2 data is a trial-centric benchmark where a set of criteria for a single trial are matched to longitudinal de-identified records from 288 patients. The N2C2 dataset is the largest dataset with real patient records. When released in 2018, most of the 47 participating teams applied rule-based criteria-specific heuristics. This brings us to the main drawback of N2C2 dataset. Given the small set of criteria used, it is prone to criteria-specific heuristics, making the methods difficult to generalize. Multiple RAG methods that evaluate on this benchmark also apply manually crafted criteria and specific prompts. We move to the text retrieval conference or TREC tasks on clinical trials. Here, the benchmarks take a patient-centric view and match a single patient to multiple relevant trials from a trial registry of 500,000 trials. Here, the direction specificity affects the number of patients involved, which is close to 50 on average. Each patient has several hundreds of trial relevance judgments. We find that the use of transformers evolving across the years from query reformulation and representation to relevance prediction. Next, we move to parsing the information. This involves information extraction of entities and relations and additionally parsing to logical queries which are applied on a patient database. Several methods apply reformulation techniques to optimize retrieval of trials. Alternatively, several methods embed patient and trial criteria using BERT and its clinical variants. Some additionally create memory networks of patients to maintain temporality in the patient records. We now move on to the matching between trials and patients. Here we formalize the matching problem where given a patient P and a clinical trial C with inclusion and exclusion criteria, a patient is ineligible if any of the exclusion criteria is satisfied, they are irrelevant if any inclusion criteria is unmet, and eligible if all inclusion criteria and no exclusion criteria are met. We see that the problem itself is direction agnostic. This is reflected in how the current LLM approaches are categorized by the task granularity, where a method can predict criterion level matches via trial level matches to rankings, by ranking multiple patients to trials or multiple trials to patients. Majority of the methods are patient-centric, where they rank trials given short patient descriptions. This leaves large room for development of fine-grained prediction methods that is of better utility to clinicians who still need to manually review trial patient pairs after a trial ranking. Lastly, we look at evaluation. All studies report accuracy metrics related to precision, recall, and F1, with the rankings methods also reporting mean reciprocal ranks and NDCG scores. Very few studies report other metrics, the most common being deployment metrics such as latency, API cost, and workload reduction, and human evaluation of reasoning quality. However, the rubrics used varies from one study to another. Given the popular use of chain of thought reasoning and LLM justification elicitations, and the lack of standard reporting of these errors, we introduce an error taxonomy based on the root cause of the errors and possible mitigation efforts. The first are the information isolation errors, where the LLMs misscope or use old incorrect scopes related to entities and relations. These could be mitigated by placing consistency checks. Next come the reasoning errors due to a gap in information. These kinds of errors usually arise in confidence miscalibration that could be better calibrated. Finally, we have the inconsistency errors, where there is a contradiction between the explanations and the predictions made. These errors allude to bias shortcuts in the LLMs and indicate the need for robust fairness tests. Thus, critical challenges still remain in the adoption of LLMs in clinical settings. We need stronger benchmarks with standardized evaluation that go beyond accuracy. We need methods that incorporate the contrastive effects of inclusion and exclusion criteria into the task learning. We need fairness and bias studies to measure the impact of the developed methods on diverse demographic slices. Finally, we need more public patient data under appropriate usage policies to test methods for real use cases, and strong privacy safeguards such as patient de-identification and digital twins to prevent data leaks and dual risks. While current methods focus on a linear recruitment pipeline, we identify the following future directions where LLMs can make the recruitment pipeline more interactive. LLMs can assist in future trial search, systematic reviews, and trial design by incorporating joint representation of trial and patient data models, and trial structure. LLMs can assist in collaborative trial planning by automatically identifying criteria and assigning them to different strategies such as reasoning, comparison, search, or deferring to an expert when uncertain. In conclusion, we looked into how LLMs can streamline a time-consuming clinical process of trial and patient matching by investigating the 4 components of the recruitment pipeline. We assessed clinical challenges in current benchmarks and evaluations that fail to assess transparency and accountability in these methods. We saw an example of how standardized error reporting can help develop appropriate mitigation strategies. Finally, I showed how LLMs can be used to make the pipeline more interactive via trial design, search, and collaborative task planning. Thank you for listening.